Hi, my name is Jessica DeSanctis, and I'm a neuroscience and biology double major. My honors thesis is titled Thermostability and DNA Binding Function of UVF and EnHD Chimera, EdUn. My project revolves around combining the functional DNA binding elements of the protein EnHD with the thermostable elements from the protein UVF. The goal of my project was to create a chimeric protein that is both functional, meaning it binds to DNA, and thermostable, meaning it does not denature at high temperatures. Adding thermostability to functional proteins is beneficial for many industries, including, but not limited to, the pharmaceutical, manufacturing, and transportation industries. Protein stability is imperative for safely handling and storing biopharmaceuticals. Denatured proteins can cause pharmaceuticals to be ineffective and possibly dangerous to the users. Additionally, to manufacture different compounds on a large scale, they need to be able to withstand the heat associated with mass production. Thermostable proteins also make transportation more efficient and cost-effective by eliminating the need for industrial freezers or trucks or airplanes. I studied this by first helping to express the chimeric protein named EdUn, which contains the hydrophobic core of UVF, which is thought to be important for thermostability, and 16 surface residues of EnHD that have been shown to be important in DNA binding. EdUn was designed by our computational team and seemed to be thermostable in molecular dynamic simulations. In similar simulations, EdUn had more contact time with DNA than UVF, but spent the majority of the simulation unbound from DNA. This indicates that EdUn is unlikely to bind to DNA and unlikely to be functional. We expressed EdUn by culturing it in E. coli with ampicillin, then lysing the cells, then purifying and concentrating the protein. Next, we analyzed the functionality and thermostability of EdUn using circular dichroism, or CD, and electrophoretic mobility shift assays, or EMSAs. From the CD melting curves, we found that similar to UVF, EdUn is thermostable and has a melting temperature greater than 95 degrees Celsius. Additionally, from our EMSA data, we found that EdUn does not bind to DNA and therefore is not functional. Overall, this data demonstrates that we can create thermostable chimeric proteins. However, more data is needed in order to create a functional chimeric protein. These tests also demonstrate the accuracy of our computational models, which correctly predicted that EdUn would be thermostable but not functional. Thank you.